Unlocking the genetic secrets of bipolar disorder. A giant leap forward. Imagine trying to solve a puzzle with millions of pieces, but you only have a few hundred to start with. That's what researchers have been facing in the quest to understand the genetic underpinnings of bipolar disorder, BD, a complex mental health condition that affects millions worldwide. But now, thanks to a groundbreaking study published in Nature on January 22, 2025, scientists have made a massive leap forward, uncovering 298 genetic loci linked to BD, 4 times more than previously known. Led by Kevin S. O'Connell and a global team of researchers, this study is the largest of its kind, involving over 158,000 individuals with BD and nearly 2.8 million controls. It's a game-changer for understanding this often debilitating disorder. Bipolar disorder is no small problem. It's a leading cause of disability globally, affecting people's mood, energy, and ability to function. Despite its high heritability, 60 to 80%, the genetic roots of BD have remained elusive. Previous studies identified 64 genetic loci, but this new research blows that number out of the water, identifying 298 loci through a massive multi-ancestry genome-wide association study, GWAS. The study included participants of European, East Asian, African American, and Latino ancestries, making it one of the most diverse genetic studies of BD to date. The researchers didn't just stop at identifying these loci, they also pinpointed 36 credible genes that play a key role in BD's development. These genes are involved in critical brain functions, such as synaptic activity and the development of GABAergic interneurons, which are essential for regulating brain activity. This discovery not only sheds light on the biological mechanisms behind BD, but also opens the door to potential new treatments. The study's key findings are nothing short of revolutionary. By analyzing data from clinical, community, and self-reported samples, the researchers discovered that the genetic architecture of BD varies depending on how patients are diagnosed and their specific subtype, BD type 1 or 2. For example, BD type I, characterized by episodes of mania and depression, showed higher heritability than BD type 2, which involves hypomania and depression. This suggests that the way BD is diagnosed and classified could influence the genetic signals researchers detect. One of the most exciting discoveries was an ancestry-specific genetic variant in East Asian populations. This variant, which isn't found in other ancestries, highlights the importance of including diverse populations in genetic research. Without this diversity, critical insights like this would have been missed. The study also revealed that BD shares genetic links with other psychiatric disorders, such as schizophrenia and major depressive disorder. However, these links vary depending on the BD subtype and how patients are diagnosed. For instance, schizophrenia showed a stronger genetic correlation with BD type I, while major depressive disorder was more closely linked to BD type 2. These findings could help refine diagnoses and tailor treatments to individual patients. Of course, no study is without its challenges. One limitation is the inclusion of self-reported data, which can be less precise than clinical diagnoses. This may have introduced some noise into the results, particularly for BD type 2, which is often harder to diagnose accurately. Additionally, while the study included diverse populations, some groups, such as African Americans and Latinos, were underrepresented compared to European and East Asian participants. This highlights the need for more inclusive research to ensure findings apply to all populations. Despite these challenges, the study opens up exciting opportunities. The identification of 36 credible genes provides a roadmap for future research into the biological mechanisms of BD. For example, some of these genes are involved in synaptic function and GABAergic neuron development, which could be targeted by new drugs. The study also found enrichment of rare genetic variants in BD cases, suggesting that both common and rare genetic variations contribute to the disorder. This convergence of genetic signals could lead to more precise treatments. The implications of this research are vast. For starters, the 36 credible genes identified could become targets for new medications. Some of these genes are already linked to processes that existing drugs, such as lithium, affect. This raises the possibility of repurposing current medications or developing new ones that target these specific pathways. Moreover, the study's findings could improve the accuracy of polygenic risk scores, PRS, which estimate an individual's genetic risk for developing BD. While current PRS aren't yet precise enough for clinical use, this research brings us closer to that goal. In the future, PRS could help identify individuals at high risk for BD, allowing for earlier interventions and personalized treatment plans. The study also underscores the importance of considering ancestry and BD subtype in genetic research. By including diverse populations and refining diagnostic criteria, researchers can uncover more precise genetic links and develop more effective treatments. This research is a monumental step forward in understanding bipolar disorder. By uncovering the genetic architecture of BD and identifying key genes and pathways, scientists are paving the way for better diagnostics, treatments, and ultimately, improved quality of life for those affected by this condition. While challenges remain, the opportunities for progress are immense. As we continue to unravel the genetic mysteries of BD, we move closer to a future where this disorder can be managed more effectively, and perhaps even prevented.